Well, and here we go again. Hi, hello everyone. I'm Oliver, and welcome to another Saturday live stream. I mean, I did a little bit of math today. I counted all of the episodes together, and I discovered that it's already. I've been doing this for 3 years, so around I don't know 150 or even more episodes. I'm happy that you could join me again today. I'll do, as always, a little bit of housekeeping at the beginning, hoping that you're able to hear me properly. And everything seems to be working. Okay, yeah, like every week, I would like to say hello all around the world, to North Carolina, United States, to England, the Netherlands. I'm reading off basically what I see over here in the chat. Of course, Norway, Oregon, Connecticut, von Vietnam, Upper Austria, London, Florida in the US, UK, Netherlands, England, as always, Poland. Okay, you can hear me well. Help from France, from Scotland. Very nice. Always good to know that there's so many people around the world who like microscopy. Okay. So what am I gonna do today? I've been Los Angeles. I just see like every week. I'm thinking a little bit of well, what should I actually be showing you? And I just realized I have not been doing oil immersion microscopy. Oil house dust. Yeah, I think I got some house dust over here. Thank you for reminding me. So basically, the idea is I've not been doing some oil immersion microscopy for quite some time now. It's because it's not one of my favorite ones, because it's a little bit messy and so on. But I thought, well, actually, I do want to get a little bit of oil immersion microscopy done as well. So we'll go up today up to 1,000 times and just have a look at some of those specimens that I've got here. As always, I'm quite open for any suggestions. Yeah, house dust is now also on the list. It does not always mean that the things that we're looking at are always very exciting. Sometimes not so exciting, but that's a little bit part of the hobby. So what do I have here? Some of the specimens over here you already know. This is some skin cells from a keyboard, right? You see some hair and so on when you turn the keyboard around. Some anthers over here with pollen. 2 weeks ago, I put it also under the microscope. Unfortunately, I was not able to germinate them. I tried to do that, but they seem to be a little bit old. And over here, I've got some fungus. Okay, some spores of a fungus. Over here, I've got a glass jar with some protozoans on here that we can go at high magnification. Yeah, there are a few things, and we're just going to, and we were just gonna see how where we're going now. There is always a little bit the challenge when doing live streams or generally microscopy videos like this. The challenge is that different people have different knowledge and experiences. So what I would like to do, therefore, is I'm just going to always start explaining it almost like from the beginning. So yeah, apologies if you already know some of the things here. Okay. However, there is one little thing that I want to show you, something that I bought myself a few days ago. I'm just going to move this away. I'm going to give you a little bit behind the scenes look as well. It's not sponsored. I'm not affiliated. Okay, so this is not an advertisement. But one of the nice things is I found that this little thing over here. It's called Stream Deck, and you see there are buttons over here that I could program. And now I don't have to use a mouse to click when I want to change scenes. But for example, if I want to switch on the microscope view, I only have to push this button over here. Look, I press it. And then, yeah, the microscope view goes in place. Or, yeah, I can also turn the arrow on and off by pressing the button, and then I'm back here at the desk view. Yeah, so that's basically, I hope that this is going to make live streaming, a little bit easier. Yeah, yeah, you've got the big one. I've been thinking, should I get a larger one or not? And then I said, no, I just want to keep it small to keep the clutter on my desk a little bit away. But you see, I can program it. I can, by going backwards and forwards, I can basically have indefinitely many keys, but I can only display 6 of them, which is enough. Yeah. So just for your info here, a little bit of some background, and actually, I think it proves to be quite nice. So I am going to switch over now to the microscope view, and right now, what we're really concerned about is the image above here. And you see, there's nothing in there, and I'm just going to explain now to you a little bit. How do you say what I'll be doing? And I also have to change now I hope I'm able to do that, the desk view as well. Let's see if I'm able to rotate around. So this is now the desk view. Unfortunately, it's upside down. I hope you don't mind. Okay. I hope you don't mind it's upside down. But what I want to show you first, before I do oil immersion microscopy, I just want to show you a little bit what the focus lock lever is, in case your microscopes don't have that, because I'll be using this. And that is, of course, the coarse focus, the fine focus over here, and coarse focus over here. And if you actually look, there is this ring out here. And what I can do is I can actually, I don't know if you're able to see this, there's this lever over here. I can push over. Okay, do you see this? Yeah, and when I push it, then it will remember the position, and I will not be able to turn it further. Okay, so it kind of it blocks it. It locks it in place. Yeah, so it kind of remembers the focus position. Yeah, and that's what I'll be. Yeah, and then I can undo it again by pushing. Where is it here? By pushing this one here again, back. Yeah, I have to be careful that my one hand is not in the way. By pushing it back, I'm unlocking it, and then I can rotate it freely again. And then when I want to remember the position again, I kind of push it forward again, like this. I'm pushing it now from the other side so that you can see it better. And then it remembers the position again. You see, it kind of blocks. Huh? So just for your info, this will be is very useful when doing oil immersion microscopy. So because otherwise I won't. It will be even more difficult, right? So not every microscope has this, and that's perfectly okay. But yeah, it's a very convenient one. Yeah. So it's a locking mechanism. It's a convenience thing. It's not absolutely necessary, right? But yeah. And in case you have not seen it yet, this is how my desk looks like. Oh, I just see. You see? There's the dust over here. Yeah, we can put that also under the microscope. Yeah, but that's the way. This one over here for your info, that's the camera that's currently recording. And then you see, of course, yeah, me over here. So just to give you a little bit of an orientation of what's going on here. Okay. So what I want to do is I'm just going to work my way through those samples here. I hope I'm not going to forget about the house dust. You know what? Why not just start with that first? And what I'm going to do therefore is the following. I have to think of a good way of actually collecting this house dust. It's probably one of the less exciting specimens. But because there's been the request, let's just use this as a little bit as a demonstration object. So, and what I'll be doing is the following. I now need a small drop of water, and I'm going to get myself. I just realized I'm again super prepared because I forgot my Petri dish or a small beaker as a water source. Here it is. And what I do is the following. In oil immersion, what's really important is you have to use a very little water. You do not want the cover glass to float, right? So what I will do therefore is I'm going to first put a small tiny drop of water on the microscope slide, and I measured it around 25 to 30 mil. Would be, not mil. Wow, I'm way off. Microliters, 30 microliters. Is 30 to 50 microliters is going to be fine. Yeah. So you see, that's not so much. We're going to see if this is going to work. And what I'm going to do now is I'm just going to use a toothpick to kind of collect some dust samples here, and then I'm just going to dip it in here. Maybe I'll find some other here in the back. Yeah, I got some here. I will be honest with you. I've been hoping to trying to find some dust mites. I wasn't in my bed, and I'm very disappointed that it was not successful. I know what you're thinking. I should be glad that I didn't find any. I know there must be there, because pretty much all beds have dust mites, but I was not able to properly extract them. So, and we already see the first problem that we have to solve. This is very thick, okay, this sample, so it's got to be thin, because we do not want a floating cover glass. So what I'm going to do is I'm going to remove some of this, the dust fibers here. Okay, that's number one. We're going to spread it out. And we drop a cover glass on top of it, and every now and then I'm going to interrupt myself. You already know the format maybe, and I'm going to read some of the comments. So if you have any specific questions or comments, you just feel free. So, and then this one goes on top of here, and and what I'll be doing as well is I'll be wiping off the excess water here on the side. This is just standard specimen preparation again. do not expect a super exciting specimen here, but it's a nice demonstration purpose. And then now let's basically start, and then I'm going to show you how the whole thing with the oil immersion works. So yeah, I'm just going to switch over now to the microscope view again. I've got it 20 times already in there. You know, normally you start at the lowest magnification here. Let's start at the low magnification. And here we go. Of course we see the dust fibers. Yeah. Sometimes these things do look a little bit like modern art. Natural fibers, of course, lots of fibers from clothing. Yeah. Maybe skin cells can always be found. So I do assume that those things here, maybe some skin cells. Skin cells are a very, very common part of house dust. Those round circles over here, of course, air bubbles. Yeah. So this is a little bit the thing. And now when we want to go to high magnification, we have to choose a certain thing that we're interested in, right? Yeah, I don't know. It almost doesn't matter. Let's see if there's something over here. Yeah, again, this is more of a demonstration here, the house dust. Occasionally you do find some interesting things like pollen grains and yeah. But I don't know which one to, that's always how do you say, the question, the difficulty in deciding. Let's just say we're just going to focus on this one over here, in this one over here. How's that? I don't know. Yeah, whatever that is. Maybe it's some skin cells. Okay. So we do the following. We go up with the magnification, of course, and we're refocusing. It's important to recenter it. And I'm going to turn off the arrow. Yeah. So this is now 40 times. Yeah. And it's quite okay. By the way, in case you're wondering about the blue color, yeah, I've got special microscope optics, TIC. If you want to see how it looks in bright field, we can also do that. That would be now the way that most of you would probably be seeing it. We can stay like this if you want to. 60 times, of course. Yeah. You see that there is a little bit of movement going on here. Yeah. Sometimes if you see those wiggly things here in the background, then these wiggly things here, yeah, depends. It could be they could be wiggling because of so-called Brownian motion. So there's this random movement of particles. Yeah, could be bacteria as well. Difficult to say now. Yeah. So normally what I recommend is if you have a microscope with an oil immersion, 100 times oil immersion objective, and normally I would recommend is take it out and replace it with a 60 times non-oil immersion objective. The reason is because it's either easier to use, and in many cases you don't need the higher magnification of the 100 times, right? So yeah, because you've got to be a little bit careful, right? Because otherwise, if you get this immersion oil on the non-oil objectives, you've got some cleaning to do here. So and now I'm now going to switch over here. Just have a look from the 60 times. By the way, even at the risk of repeating myself, but there are always a few new viewers here. So if you actually look at the top here, right? There's this ring here, and this is and this one is a nice one because I can actually turn it over here. Yeah. And the question is, what does this do? This is a cover glass thickness adjustment. So you see right now it says 0.17 here, right? Yeah. So over here you can adjust for different thickness of cover glasses to get the best resolution. I rarely use it, but just for, yeah. So, and now I'm going to rotate the 100 times oil objective into place. Now look what happens. I rotate it in place and it looks kind of blurry. Yeah, it's out of focus, so let's focus a little bit. So, and this is how it looks like. Okay, let's center it again. Okay. Let's center it again. Over here you see that in the background there's also some dust and dirt, but this is without immersion oil. So even though we are able to see something here, it's not quite as crisp and as clear as it could be. Yeah. So this is the thing here. There are again a couple of questions here. Is it an internal aperture or does it shift a lens or up and I don't know honestly what the ring actually does. If it's yeah, I don't think it's aperture really, because if it were aperture then it would also change the brightness, I would say. Yeah. Which it doesn't do. So I suppose we were talking about this ring now of the previous objective, right? So and now I'm going to do the following. Now you gotta take. I don't know. You know what I'm gonna. Shall I try something? I'm gonna try something now. Just a second. If this is possible, I'm going to try to duplicate this. Just a second. Okay, I duplicated this, and let me see if I'm able to do the following. Look, am I able to move this? I'm able to move this. Isn't this cool? Let's make it bigger. This is actually nice. I never tried this actually here. Okay. Maybe this is actually nicer to see it better like this, what I'm doing. Maybe I'm not gonna, yeah, we'll see. Okay. Maybe I can actually stretch it out over the whole image here. How is that? So yeah, maybe a little higher here, like this, and then maybe I can stretch it down over here a little bit more. Yeah, the OBS Studio is the program. It's a pretty nice one. Yeah, so I think that should be okay. Yeah, let's leave it like that. Okay, and now I'm going to. Now you can actually see much better what I'm doing. Yeah, zooming into the microscope itself. Yeah, so I'm going to now do the following. We've seen that everything was in focus, okay? So let's have a look here. It's in focus, right? So what I'm doing right now is I'm now engaging the focus lock lever that I showed you before. So I'm pushing the lever over. I'm pulling it towards myself. Okay. And that's it. Now I'm doing the following. I use the coarse focus, and look, I'm actually lowering it now. I'm lowering it. The camera is a little bit slow that I have here. That's why it's not quite as smooth. I can do away with the arrow. No, how do I do it? The arrow. Ah, I see. That's this one. Yeah. Here I have to take the arrow away. And now I'm going to put maybe I turn it down a little bit so that it's not quite as bright. Yeah. Yes, it's maybe you see it better. And now I'm going to put a small drop of this immersion oil. Okay, look, all the warning signs on here. Yeah, it's an original Olympus immersion oil, and it's clear. It's a synthetic one. Many immersion oils that you get these days are actually yellowish, but this one is completely clear. Yeah, so and it will stay clear. And I'm going to now put a drop directly over here on the cover glass. Okay, and now the fun part starts. Because what I'm now going to do is because I engaged the focus lock lever, I can turn the coarse focus all the way up until it stops, and it will not be crashing the objective lens into the slide because it remembered the position. And now we're gonna go over to the other side here. Turn on the light here. And I have to, see it got a little bit out of focus. I move it back and forth a little bit to spread the oil. And then I focus. And I think you see that the image is significantly more clear, okay, than before. Yeah, it's also brighter. I mean, but that is essentially what basically this was. Look, there's some water evaporating. Yeah, and there's an important question over here. Had a yellowish one, do you mean that it was made naturally instead of synthetically, hence the color difference? Yes, that's correct. Those yellowish oils are basically natural immersion oils. I don't know what they use. I think they call this camphor oil or something like that. They do have a disadvantage, those natural oils, and that is over time, they will actually turn hard and solid. Because there are some kind of some how do you say, substances, volatile substances in there that will evaporate. and then over the years it can actually turn hard. The idea is that if you use those natural immersion oils, you should clean the objective afterwards every time to prevent this from turning hard. However, I've heard some people actually also say and read it online that people who are doing a lot of oil immersion microscopy, they don't always clean the objective right away after they've used it, because they just keep the oil on it and they just continue the next day, because it's always in constant use anyway. So that is basically the thing. So that's basically dust. I mean, we can move around a little bit. The oil will spread. Yeah. Of course, I don't know where we can now have a look at. So many of these things indeed seem to be, I don't know, skin cells. I don't know where the fibers now were. I have a difficulty finding this now. But I mean, you get the idea. You're definitely most likely skin cells. See, that's one of those. Here we are now with those fibers. If you want to, we can try to put it with DIC, which of course uses more light. I open it a little bit, and then we have a little bit of some nice color as well. Yeah, but look, just by tapping my table, you see that there's this vibration, because of course everything is magnified, all of the vibrations. Yeah, and so that's basically Oh, I don't know what this is, for example. This weird thing. Yeah. So that is about cleaning. How about cleaning it? I'm going to show it to you as well. I'm going to be using not tissue paper, lens paper. Yeah. Why are the moving particles going the same direction? That's correct. Usually this is when there is a streaming motion of particles, then this basically means that there is because of capillary action, the liquid starts to spread and it also starts to evaporate. And it could be that because of the dust fibers and so on that are in there, that the cover glass is not completely flat, but it's maybe tilted a little bit. Also, it is like this, that the specimens, like the fibers that you see, they slowly start to absorb water as well. And this kind of also causes them to swell maybe and to deform. And depending a little bit on how the stresses are beneath the cover glass, this is basically how the water will then move. So this is something that it's an artifact we simply ignore. And sometimes if there's some tiny vibrations, like I don't know if you're able to see this one here right now in here, this dot here, right? It does not always mean that it's moving on its own. It could be so-called Brownian motion as well. Bacteria that move, they have a very distinctive motion. It's way more directional, yeah, and it cannot be mistaken so easily, right? So yeah, I think I'm just going to move on because we still have a couple of interesting things to look at. So what I'll be therefore doing is kind of remembered the position right now, yeah. So I'm going to now, lower the stage again. You see everything will go out of focus, right? I will remove the slide and basically I'll put it somewhere else, and we just have a look at the next specimen, and I will just now use again this the scene switcher, and we're just going to have a look at a few of these things. By the way, just for your info, something you did not see yet is this is also some yeast with sugar, which I put again in my coffee warmer, hoping that 1 hour ago, hoping that we're able to see some yeast cell divisions. I don't know. We might still have to wait a little bit for that. So, and we're gonna do the following. Same story as always. Let's have a look at a few of these things, just because it's kind of fun. I mean, we already looked at skin cells, so why not just continue doing that? So I'm going to now use those here. Here, that's enough. How am I gonna get those? You know what? I'm gonna do it a very easy way. I'm just gonna pick up some of this. Generally, I think this is kind of very large. Okay, those specimens are very large. Yeah. You see it here on the bottom. I think it's a lot. Cover glass goes on top. Yeah, and because some people keep on asking me what do I do with those slides. I washed them. I just recently washed them. What I did is I You see, it's so thick, it's not even fully. Yeah. If I cannot press it down completely. I washed them. I put some dishwashing detergent from a dishwashing machine, the powder on it, and yeah. So this is actually something that's not so good because you see that it's not flat on here. Yeah. And why is this not good? Because then it's so thick it's going to be difficult to focus, and it's also not entirely stable. I'm going to remove this again, and maybe I'm able to take out away some of the larger pieces here. Not a lot better. Not a lot better. So what I'm going to therefore do is I'm just going to add a little bit more water here on the side, just to make sure that it's completely filled up, or more or less completely filled up, because the water will actually stick the cover glass to the slide. And I will remove here again the excess water. Yeah, it's a little bit thick, the whole thing. And what we can now do, it's a little bit faster. I love the switching thing, so much faster. What we are now able to do is the following. I'm able to now directly, I'm going to center it a little bit. I'm able to put now directly some immersion oil on it. And I don't have to go through the lower magnifications because it kind of remembered. Yeah, so I'm turning up, turning up, turning up, turning up, turning up, up, and spread it a little bit, and then hope that we're able to find. See, but the focus I've lost. Ooh, look! Did you just see the whole thing whizzing about? Yep. This is here a bubble. Okay, so now I have to find those little, but anything that we find, we put it into focus. Oh yeah, there's something interesting here. No, actually not really interesting. I think I did not catch a lot. That's here are a few. Yeah. Very nice. That is what I wanted to show you. Very nice. So I need to explain this a little bit here. Yeah. The cover glass moved. Yes, this indeed is something that we also sometimes see. Sometimes when you're focusing, because the oil exerts some pressure. Just by turning the focus a little bit, you can actually move the specimens around. But this one over here, I just need to explain because I think it's kind of nice looking, but it doesn't make a lot of sense if you don't know what you're looking at. So I looked at those skins that kind of were shed off, maybe some dandruff and so on. And what you're able to carefully, I don't know, let's turn this down a little bit. No, it's too dark. But maybe you're able to actually see like different, it's almost like different plates that are overlapping here, right? You can actually see very, very lightly, those irregular. Yeah, so basically these are the individual cells that are kind of stacked next to each other. Yeah, and so it's not only the cells are still connected together because it's the epithelium, the surface of the skin, right? The cells are also together, and if now a larger skin area breaks off, then the cells themselves are still connected to each other. So what we're actually seeing here is we're actually seeing if you unfocus it a little bit, you can actually see the surface of the skin. Yeah, how it looks like. Of course, the individual cells are difficult to see separately because they're all connected, but maybe carefully a little bit, if you defocus it, you can actually see, okay, that's here is one cell, then over here, the borders of the cell, like almost like different plates arranged next to each other, right? So we're looking at it directly at the skin here. I'm going to see if I can move it all the way over there as well. I don't know what this is. This one is already way too thick. Yeah, cell tectonic plates. Yeah, but another good one over here. Maybe over here again. Yeah. Do you see this thing moving here? Do you see this thing here moving? This is not Brownian motion. Somehow. This is also quite large. This does not look like... I wonder if this is not... This jittery movement... Yeah, could be some kind of a microorganism. I think Brownian motion is still... Brownian motion looks even more random than this. Yeah. Okay, so that's basically some dandruff, so to say, and some skin. And look what I'm doing right now. Do you see this one over here moving back and forth? Okay, look at this here. Okay, look, and now I'm focusing. As I'm focusing, it's moving back and forth. Why is that? And the reason, again, as I told you before, is because the oil over the objective is exerting some pressure on it, and this kind of pushes the water back and forth. Yeah. So this is not because of its that's not the natural movement, back forth. Yeah. See. As I'm focusing, and so that's also an artifact. Yeah. But honestly, it looks very irregular for a microorganism, if you ask me. Yeah. So let me see. Yeah. So again, I think we're going to have a look at I'm going to have a look at another one. So these are basically skin cells. I think a little bit too large, maybe for oil immersion. So let's go on to the next one. I'm just checking the time a little bit. As always, very easy to do now. I'm just going to lower it again. Yeah, and take it out. So you see that this focus lock has this really nice advantage that you save yourself quite a bit of time because you don't need to go through the lower magnifications and everything. So let's put this away. What do I have here? I don't know. This is still good. Okay, I'll have a look later. You know what? Why not? Why not try it now? I mean, this is a Petri dish. Where it's already a little bit dried out here. This is basically, these are some cells that are isolated from a cheese, because cheeses, of course, have yeasts and bacteria growing on them, some of them, and I basically isolated that, and I was growing it on this Petri dish. What I would like to do, however, is the following. And these are not bacteria discovered, or not purely bacteria, but also yeast. But there's this contaminant over here. This is the yellow colony here. I don't know what this is. Okay, so with unknown microbes, you always have to be a bit careful, obviously. So what I'm going to do is I'm going to take a small sample here. Gonna put it or spread it over here. Normally, you have to be very careful that you don't take too much of it. Okay. What about oiling the condenser? Never tried that. I know that there are. Because I've never. Sometimes it's possible to actually put an oil immersion, an oil drop directly on the condenser as well, so that there is oil on the bottom and also on the top. So there is basically no air gap in between. However, I'm not so sure about my microscope to what extent this is actually also oil proof or not, therefore, I also don't want to have to spend a lot of time cleaning it up later on. So for this reason, I never tried putting oil on the condenser lens. Yeah. So we're gonna give it here another try. Okay, so this is the unknown contaminant from the Petri dish. Okay, I will put this here away again. Let's wipe off some of the water, and here we go. Again, I have not tried this before, yeah. So as always, there is always a small amount of uncertainty. So I need to change over to this here. I'm going to put a little bit, and I hope I have enough of the cells there. I don't know. So let's turn it up. And yes, bacteria. Yeah. You see that it's kind of very dark at the surface, so maybe the oil is not completely spread. Yeah, see, was a bubble in here. I got a bubble in here. Yeah. And as we can see, it's not even a so-called pure culture. You see that there are different, the bacteria have different shapes as well. Yeah. So these are basically bacteria, as how do you say, as expected, bacterial colony. And yeah, a lot of people, especially beginners, they are really curious. They want to see bacteria under the microscope, and what microscope do you need? And I will say, yeah, but actually they are not the most interesting specimens. This is in DIC. Okay, normally you want to look at bacteria in phase contrast. Now I do not have a 100 times oil phase contrast. Do you see this rapid movement? That is usually when some air bubble, yeah, pops somewhere. So what I'm going to do now is I'm going to remove the DIC. This is bright field to bright. And this is now basically how it would look like in bright field with the condenser all closed. Yeah. So. And also here, see, if you move the focus, then it will actually move the liquid and the bacteria. I have a yellow lamp in my microscope. How can I filter it to white without changing the lamp? You use a blue filter. You definitely use a blue filter. If you're using the old tungsten or even halogen, then they have a significantly larger red component. And there are some microscopes already have a built-in blue filter, or if not, you can maybe in eBay. In the days of analog photography, there used to be the so-called daylight filters that converted the color, and it was bluish. So yeah, that's it's fairly simple. You just put the blue filter over the lamp, yeah. So or into the condenser filter holder, and this will basically not make it appear blue, but it will convert the reddish, yellowish, orangish light into a much more neutral white. Highly recommended. Yeah. Oh, nice comment. I've been watching your channel for 8 years now, and your reason I'm still in this hobby. Thank you very much. Yeah, I started this channel back in January 2018. Maybe some of you who have been following around even longer. I've been before that. I've been for a couple of years. I made some basically PDFs, microscopy PDFs for download. If you want, you can still download them from the webpage, from the website. So people sent me articles, and I did the formatting and a little bit of editing, and then kind of made a layout, and then I made it available for download. I did this for, I think, a couple of years, but this was a lot of work, honestly, doing all of the... Yeah. The reason, I'll tell you now the reason, maybe, as we watch those bacteria, I can tell you a little bit the reason of why I wanted to start this PDF microscopy magazine, because at that time, just like right now, I was very interested in astronomy, amateur hobby astronomy, watching the stars and the planets, bought myself some telescope and so on, still interested. Maybe not as active as I used to be, but usually in the magazine shops in the train station and wherever, there were usually always some astronomy magazines available, but nothing about microscopy. So I said, gee, there should be also microscopy magazines. And so I decided, why not just try it? And people send me articles, I do the formatting and the layout, and just make it available for download. Yeah. This was a lot of work, however, and then that's why I decided to start the channel, Microbehunter channel, not this one, the other one, the sister channel, and then which was mostly about was about microscopy, and then shortly afterwards I started a technical channel, which is this one that you're watching right now, where I talked about optics and did some viewer Q&As. And a couple of months ago, I tried to sort out the channels a little bit better, so I pivoted the other channel. I'm doing more like a theoretical biology, microbiology explainers. Currently about food safety. You might be wondering, why am I doing so many food safety videos on the other channel? And the honest answer is is this because people watch it? Why does food spoil, and how do tin cans preserve food? This was one of the most successful videos recently, got over 200,000 views. And people are interested in that. That's fine. But I then decide, okay, the real microscopy stuff and the hobby microscopy and observation, it should also have a platform, and that's why I kind of moved those videos and live streams over to this channel where you're watching right now. Yeah. So just so that you understand a little bit how it came about. Yeah. Ah, yes. Yeah, astrophotography I've been also doing. At that time still I developed my own films even, so I took pictures analog. But these days with a good lens and a digital camera, man, you can do a lot in astrophotography. However, the thing is I consider microscopy more, in the sense, more convenient because you're not so weather dependent. You can basically always do microscopy regardless. And where I'm living in Central Europe, the weather is not always very stable, right? And so for this reason, I decided some time ago to place a stronger focus then into microscopy, which also kind of matches a little bit my education that I have, because I studied microbiology at university. Obviously, you know that already, and as a teacher, sometimes I also use those videos for demonstration purposes. Yeah, so these are unfortunately pollen which were a little too old. I say unfortunately a little bit too old because I could not get them to germinate. Okay, I tried. It didn't work. So I wanted to grow pollen tubes. Yeah. Permanent slide off onion skins. Ah, good point. You know what? Before I do this, really important announcement. I almost forgot. Next week I have to skip the live stream. Okay, unfortunately, because I'm not here. That's the only reason. So if you're kind of wondering why I'm not doing a live stream, it's simply because I'm not here. I have to go somewhere, and over the weekend, and for this reason, it won't be possible to do a live stream. Just so that you know that I didn't just quit doing live streams. Okay, so. Yes, a chess match. Yes, you're correct. Yes, that's correct. I've not me playing chess, but I've got two boys, and the younger one I have to bring to a chess competition. Yeah, and so he has to play 2 rounds, and it's a pretty important one, so I'm jumping into the train with him on Saturday morning, and I'm going to stay until Sunday, and that's the reason why I'm not here. So let's do this one, the next one here. Get my robotic chess set. Seriously, are you? Did you order one? I wonder how well they actually work. Yeah. Next Saturday we can go. You can go on a microscopy party. I mean, in astronomy they have those star parties in some countries where you kind of meet up and you talk about, you watch stars together and so on. Would be kind of nice to have this also in microscopy. But then again, at least these days with the internet we can at least do it in a virtual way. So I didn't show you this to you. I put it in now. So you see over here, this is kind of the objective, not in contact. So you see there's some kind of an oil drop still on here. So watch again. You know what? Can I not do it like this? Just a second. Boop. Here we go again. That's what I wanted to show you. So I'm not quite in the center, because in the center, I think maybe it's gonna work. Right. So let's put again a drop on here. Okay. Yeah, my talk about chess, my son, actually, he really wanted to have a robotic chess as well. But I told him, the cover glass is moving a little bit. Yeah. But I don't know if it's possible to play speed chess. Yeah. So, we have a focus. Look at this. No, I see, it's kind of... do you see this? This is kind of like an air bubble passing by, or the oil film readjusting somehow. And now it's better. Look, it's almost the pollen are kind of rolling. This is huge, right? Yeah. So these are the pollen grains of a flower, of a tulip. And you can now also try DIC, a little bit brighter. Yeah. And my hope was a little bit, if you add sugar solution and you have to also add the correct minerals and the pollen is not allowed to be too old, then what's going to happen is that they, after, I don't know, half an hour or so, they will actually start to grow a tube on the side here. Yeah, and in this tube, you will find the sperm cell to fertilize the egg, the ovum in the flower. And you can try to induce that by adding a correct concentration of sugar solution, and you can actually watch the pollen tube grow under the microscope. But the setup is a little bit delicate, and I was not able to manage this yet. Yeah, yeah. So, I see. No, I'm just reading the comments here. So that's basically the pollen, and yeah, let's have a look at another one. I've some time ago, I've also maybe during springtime, I'm going to do another pollen session where I'm going to be maybe extracting some pollen from honey again with a centrifuge. That's quite fun as well. You can actually see the different types of pollen. However, the study of pollen is a pretty advanced field, to my knowledge. Many pollen grains do look similar as well. But by looking at the pollen, you can actually, some folks are doing some forensics even, or are able to determine from where the honey is from. Yeah. Yeah, so I'm just reading occasionally. Every time when I'm a little bit quiet, then I'm reading the comments here. Yeah. Yeah, so these are. Now the problem is the following, and you have to, when you're using oil immersion, you have to kind of restrain yourself from doing that. Right, because what you should not be doing is right now, I want to see the whole thing in lower magnification, right? And then what you do is you rotate the objective, and then essentially the non-oil objective also gets dipped in oil, and then you have to do some cleaning, and that's not so cool. And there is this anecdote that I occasionally share, which I thought was funny, but it's not funny. It's more like a it was a telephone call a couple of years ago when I bought the microscope. I talked to the representative of the Olympus company. No, it's not funny actually, but it's kind of it's a very human thing, and that's why I kind of liked it. We were talking a little bit about, because I was buying this microscope, and we chatted a little bit, and then what microscope, because I'm a teacher, what microscopes are you using in school and so on. And then I kind of said, we took away the oil immersion objectives, and we are not giving the student any immersion oil, because what's going to happen is they're going to then start dipping the non-oil objectives into the immersion oil, because the students simply are still learning, right? But then the objectives become dirty, damaged maybe, and then you cannot use them, and then you cannot use them for education. You cannot, and then the students are frustrated. So we said, it's okay. We're going to take the oil immersion objectives out. We're not going to give them immersion oil, and we're just going to do regular microscopy without immersion oil. And then the representative of the company started to laugh, and he says, "Yeah, it's not only students." It's not only students. And I said, okay, tell me about it. Yeah, we just have this just sent us an objective to be repaired. Yes, repaired and to be cleaned, because it was they got immersion oil behind the lens, so the oil seeped, because they used a non-oil objective, a very expensive one, and they cost thousands of euros or dollars, one just the objective, highly specialized ones, right? And they got somehow oil, because the person who used it in the lab didn't use it properly and got immersion oil, and the oil got soaked in, and it was even inside the objective, and so they had to disassemble it and get it cleaned. Yeah. Now the story here is the message is things like this can even happen to experienced people, right? If you're careless and you just rotate it over, and then and the other thing is those very expensive objectives, they can be serviced. Yeah, and they can be taken apart and cleaned. Yeah, so yeah, just saying. Yeah, that's why I'm deliberately careful with oil immersion, and in many cases you don't need it anyway. Do you see those bubbles over here? And I wonder what they are. And these are, yeah, I wonder if these are actually some air bubbles on the surface, or if this is some kind of I don't know, the cell membrane bulging outwards. Yeah. So that's the question. Yeah. So what time is it? 50. Oh yeah. I still have a few things I want to show you because I didn't yet get to the protozoans. So let's do some more. Let's take it out again. You know what? No, not the protozoans over here. Let's try this here. This here is, I also need to explain it to you. I also, some months ago, I started this. This here is something that you probably don't want to have so much because these are actually, that's a mold from an orange and a lemon. And what I have done is the following. Okay, I'm just quite easy to do this yourself. However, yeah, you got to be careful. You don't want to inhale this stuff. So what I've done is what you see, this white paper here in a Petri dish, that's a coffee filter paper. Yeah, just basically, I don't use commercial filter paper for cost reasons. I just use a coffee filter that you get by the supermarket. I cut it out in a circle, and then what I've done is I pressed some orange juice into the filter paper so that it got covered in orange juice. Not swimming, anything, just yeah, it's wet with orange juice. And then I took a how do you call this, a toothpick, and I scratched some of the mold, this greenish mold from the orange or the lemon, and then I kind of spread it over here. And 2 or 3 days later, it started to look like this. Yeah. At the beginning, you don't see the gray spores, but later on you do. And however, what I tried, and that's also interesting, I tried to subculture it. So I tried to take some of the spores from over here and put it again on another Petri dish, and it did not work. Because the spores were already too old, I guess. Yeah, so that's also, I found this also kind of interesting. I thought that those spores are quite resistant, maybe, to drying, but at least in my case, it wasn't like that. Yeah, so I took from this old sample and I transferred it to a new one. It didn't grow for whatever reason, which kind of surprised me, right? So this was the original one over here, and those lines that you see. Do you see those lines here? That's because I scratched off from the orange and then I kind of smeared it, yeah, over here, and this is where it started to grow first. So again, when you open these things here, those spores spread. Some people get allergic to that stuff. You don't want to have it in your refrigerator or so. The reason being that, of course, it can infect other fruits, and you don't want that. Yeah? So I'm going to put again some water on here. Okay, let's have a look at a few spores. I dip it in here maybe. I should have used a fresh one because maybe I transferred some pollen in here. I don't know from before. Maybe, doesn't matter. We're gonna see it. You see the tip is green. Yeah. So they don't dissolve quite as well as the pollen. I do get the impression that maybe sometimes those spores are hydrophobic. And let's put a cover glass again. And I'm going to now demonstrate to you. No, I can't do that yet. No, actually I can. I'm going to demonstrate to you now how you can do that without the focus lock lever. Okay? So let's try that, because many of you might not have a focus lock lever, but might have a oil immersion microscope. So we start at the low magnification. You see, this is things. I can't. Of course not. So here, these are the spores. Those tiny dots that you see. We're going up now with the magnification. Obviously, we're going to choose an area. Yeah, those spores here, we want to have a little bit larger. Okay. We have to keep working our way upwards. Here we go. It's a little too dark, so we open the condenser a bit. Here, that's better. So, and now I'm going to show you how I do that without the focus lock lever. So I've got still the 60 times objective in here, right? You can see now the ring much better that you can turn, right? Yeah, so the scale over here indicates the cover glass thickness 0.17 millimeters. So let's do now the following. I'm going to now, and this is where you have to be a little more careful. I'm going to rotate the over here like this. I apply now carefully a drop of immersion oil on here, which you cannot see because it's overexposed. But just trust me, there is a drop of immersion oil. And then I rotate the oil immersion objective into place. And then usually I go back and forth a little bit like this. Okay, it's supposed to, the camera switches to black-white when there is not enough light present. Okay, here, yeah. Here we go again. And then we have to use the fine focus. Okay, then we're using the fine focus to get everything into focus again. Yeah. Oh, I bumped it. Yeah. So, and you know what, I'm going to engage again the focus lock lever simply to remember this position better. Yeah, yeah, we can, let's have a look at bright field. And when I use the fine focus, you see, the things are starting moving again because of the pressure that's exerted on the cover glass. What I've also seen, and you can try this as well, is of course I tried to also see the growth of those spores under the microscope. This is also possible, but I've seen that the spores that are in the center of the slide, they did not grow the fungus, only those on the side, and I suppose that this has to do something with the oxygen availability. Yeah. So, yeah. So these are basically fungal spores here. Thank you. There are a couple of questions here again from you. Yeah. If you have any more questions, then please, yeah, feel free. Yeah, so that's also interesting. Not all spores look the same. Yeah. And sometimes, yeah, of course, as soon as you add water, they might actually start to swell as well, absorbing water. So let's put it this way, some interesting questions. The mold is a hazard risk, isn't it? Since we don't know what kind of mold was on the orange. As a matter of fact, yes, we do kind of know which one it is because they're only very specific ones that are on the oil. I forgot the name now. But it's a very typical one, which is actually also responsible for most of the harvest loss in citrus fruits. Yeah, it's a very well-known mold, actually. Still not healthy, obviously, to inhale it. But then again, yeah, if stuff like this is in the supermarket or in the refrigerator, we're exposed to it in any case. And for this reason, over here now in the Petri dish, yeah, I consider it quite safe because I'm not spreading this around. So sometimes when you're actually touching the fruit and so on, you might be inhaling more of this stuff. So in this case, we actually do know the name of the mold. However, I don't want to get this in contact with any other fruits, otherwise they also become moldy. Yeah? Just saying. Yeah? So the next thing, let me see. I'm just going to. Is it mostly penicillin? Some mold, penicillium, you mean? No, this specific one not. I'd have to do. Maybe someone of you can do research. The name of the green mold. Which is found on citrus fruits, then yeah, when I see the name, I remember it. It has those finger-like it has those finger-like spore-building structures. Well, mold is a fungus. Yeah, and in with fun, yes, digitatum. That's correct. Yes, Penicillium digitatum. Because digitatum means from finger-like, because it has those finger-like extensions when within the spore-building structures. That's yes, competition. Who was faster? Okay, yes. That's why for your info, this is why actually penicillin, the antibiotic, is called penicillin, because it was originally found in the fungus penicillium. But by the way, for your info, the fungus that you find on cheese, on camembert cheese, this white fluffy thing, is also penicillium. Yeah. So that is yeah, there are quite a few of them. Yeah. So we now do the following. We move on, and let me see how well this is doing here. I'm going to be trying now, and I don't know what time is it. 1 hour. Yeah, I still have those the jar. Let's do now the following, where I don't know how well it actually worked. Let's try the yeast now. The yeast with a sugar solution, and it's been now sitting here for 1.5 hours. And I hope that this is kind of yeah. Just show it to you again. You already know some of the things that I have here. Let me put this away. I can't do this. Let me lift the microphone over it. Almost spilled it. So I put it to 40 degrees, and here, in here, I've got a little bit of yeast, and I did not add sucrose. I added glucose. Apparently, this works better because glucose is a monosaccharide, so it does not have to split the sucrose into two monosaccharides. Yeah, so it's a little chemistry here. The yeast should be able to use the glucose faster than the sucrose, which is the regular table sugar. So I'm going to try it. And the idea is, and this is kind of the theory behind it, that we're able to see after an hour, after 1.5 hours, that we're actually able to see some of the dividing yeast cells. Yeah, let's see. I don't know if it's ready yet. Doesn't matter. This is called the budding of yeast. Is when the yeast cells start an asymmetric cell division. And this usually starts, according to the instructions I read, maybe around an hour after. And temperature control is very important. If it's too cold, it's not going to happen. So let's do the following. I'm going to put this away again. The spores go in here, and maybe this is a little too much water. Just let me quickly remove some of the excess fluid from the slide. Here we go. It's better. And where's my immersion oil? Here it is. Okay. So, and we go up, center it, up. I hope I'm able to find this. Oh, I've lost focus. Something here, just something here. Look, it's still swimming. Okay. Yeah, so these are now yeast cells, which is, believe it or not, also a fungus. Okay, and maybe we are able to see a few of them. Yeah, I'm going to point out using my arrow. Let's look for those cells where there is a large one connected to a small one. This is called budding. Or maybe also over here, I don't know. This is called the bud, and that's it's mitosis, it's a cell division, but it's an asymmetric one. Yeah. So over here, you can actually see a small tiny bud appearing. Yeah, so that's very typical of this baker's yeast, that they do this asymmetric cell division. Yeah, here actually quite nice, nicely visible over here. There's one, and over the course of I don't know some time, we should actually see that this bud will start to become bigger and bigger, and then we should also the cell should also divide. Yeah, so I can imagine maybe it will start to grow in size until it looks like this here. Yeah, but this one is almost difficult to see. No, 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 not all fungi have a mycelium. Yeah. Yeah, Saccharomyces. This baker's yeast is a fungus without without growing in those filaments. Yeah. And there are because the thing is the following: whether we group something together into a fungus or so, also depends, or actually does depend on the genetic similarity. Yeah. Also, for example, there are certain other features that it shares with other fungi. For example, cell wall made out of chitin and not cellulose, and so on and so on. Yeah. So there are also other factors, but of course it's the genetic thing that we're looking at. I don't know, over here, that's a big bud over here. Yeah. How do you define fungi? In the modern way, these things are defined genetically. So this means, if you go back into history, basically those creatures that are running around hunting, these are animals, and all the things that do photosynthesis are plants. This was a very easy way of classifying organisms, but then they discovered, well, there are some bacteria that are doing photosynthesis as well. Yeah. So if you pick out individual characteristics just based on shape or appearance or the way they live, there will always be exceptions, right? For this reason, nowadays the classification is based on genetic similarity. Yeah. So because and sometimes you are lucky, but sometimes you do find characteristics that essentially you can also see under the microscope. I mean, I'll give you an example here. It's a little bit because I did when back at the time at university, I had to do bacterial taxonomy, classification of bacteria. I'll give you an example. I mean, the problem or the difficulty that biologists have is and I always use a car example. You know, if you you go on a road and you see a lot of cars and you want to identify what cars are they? Mercedes, BMWs, Fords, or whatever they are, right? And then you can look at characteristics and you can say, well, I don't know, I see a green car, it's a Mercedes. Of course, you cannot make the conclusion that all green cars are Mercedes, right? So it's not a very good characteristic. However, if you see somewhere a Mercedes star on the car, well, then you've got a characteristic that's very typical that you can only find in cars of the brand Mercedes. Yeah, so it's difficult actually to just by looking at something, it's difficult to just classify them. Or you cannot classify it as an analogy again. Obviously not. You cannot classify or group cars by the number of wheels. Well, all cars have 4 wheels. That would be a characteristic that's too similar, right? And this kind of makes taxonomy a little bit difficult. Yeah. So what they are going nowadays, what is called the so-called phylogenetic relatedness. That means basically similarity based on the DNA. Yeah. Again, see those are connected together here. Also a bud that's already quite large here. Yeah. And animals that steal chloroplasts and then use them for photosynthesis, is this an animal now or a plant? Well, I don't know about. Okay, one has to be careful here. If a paramecium eats algae to do endosymbiosis, that's possible. Then actually it's still a paramecium doing endosymbiosis, right? So this is a little bit the thing. Or do you notice, of course you know ice bears. Did you actually know that ice bears are sometimes have this greenish fur? Normally they have a white fur, but under some conditions the fur of an ice bear can be greenish, slightly green. And they found out that these are actually some algae and photosynthetic algae growing in the fur of the ice bear. So is an ice bear now an animal or actually a plant? Theoretically, one could say, well, actually, It's the algae that is carrying an ice bear, or is the ice bear carrying an algae? It's just a little bit of a joke, so to say. Yeah, yeah. So, but one of the things that I like about biology and is that sometimes the how do you say our general understanding of biology of our environment is kind of sometimes yeah, questioned a little bit. One of the things that I always ask myself is the following: look, yeah, you look at those over here, right? Those two are dividing, or this one over here. There's this tiny little bud here on the side, right? The question is now the following. Yeah, normally would say, yeah, of course, it's kind of the bud over here. That's kind of like the offspring, the next generation, kind of, right? But actually, no, you cannot say that. It's genetically exactly the same as the other cell. Yeah. So just because one of them is larger and appears to grow out of the other cell does not mean that one of them is the original one and the other one is the offspring. Yeah. The size doesn't make a difference. Genetically, the DNA is the same. So Euglena, exactly, that's a classic. Euglena is a protozoan, is able to do photosynthesis and is able to feed on organic material. When scientists discovered this big confusion, is it now a plant or an animal? It's able to eat organic food like animals, it's able to do photosynthesis like plants. Is Euglena now a plant or an animal? Right? Well, it's neither, it's a protozoan. Yeah. So this is the thing, and that's what I consider also a little bit fascinating here. Yeah. So, you know what, I'm talking too much again, because let's have a look at some of those, the water sample that I've got, because, yeah, in a quarter of an hour, I want to already call it a day, because, again, I'm a little tired, but over here, you go, yes, here we go. So, let's do the following. Yes, this is, I don't know, you maybe you remember several months ago, I dumped all of the water samples that I had together into a jar during a live stream. I don't even know when this was. Was it last year, maybe? I don't know. Let's have a look. And I've been keeping the jar now. Let me put this away. Just put the coffee warmer away. And of course, water evaporates always, so I always keep on refilling it. And here it is. This is a horribly disgusting looking thing, and I love it. The more disgusting, the more you're able to see. Look at this. Isn't this? Yeah. Okay, so this is the jar. I just showed it to you from the side here. This originally had some pond water in it, and I tried to make a little mini aquarium to put some pebbles in here to increase the surface area a little bit, and so on. And then over time, stuff started to grow, and some months ago, I just dumped all of my water samples that I had together in here. Yeah, so yeah, totally. And I kind of was waiting for something to develop. And you see over here on the surface, there's this stuff. It's like almost like a skin floating, and this is a sign that there's a lot of bacteria and protozoans are living in here. Of course, there where there is oxygen. Yeah, that is yeah, and the density is not quite as high, but there are some of them are quite yeah, there are a few in there. Not extremely much, but I guess it depends also a little bit how you're taking the sample. And what I'm going to do now is I'm just going to do it like this. And I'm going to take my pipette again, and I'm going to just go in here directly in it, and I hope to get something. But maybe I don't know if this is enough really. Let's take some of this, a little bit of goo. Still stuff sticking here on the tip. Doesn't even want to come off, but you know what, it doesn't matter, because I think a lot of stuff already came off anyway. Let's do this. Here we go. Yeah, let's trash it again. I do have a little trash can over here. Do you want to see my trash can as well? No, I'm just going to show you also the trash can that I have. Let me put this here on first. Yep. So I'll put it also under the microscope. My trash can looks like this. And I know, yeah, all of those tips in here, I know it's a little bit of a plastic issue. Yeah. Yeah. But I find this still less of an issue than using, for example, yeah, I mean, sometimes I also use those pipettes here. Yeah. But then you'd have to probably rinse it after every use. So we're going to do the following. We're going to put it again over here into the microscope. I think this, let's see how this looks like. Again, drop. Okay. And let's have a look if this actually works. Oop. Yeah, again, I've lost focus a bit. Yeah, I see it's stabilizing now. As the liquid is kind of spreading. Oh, lots of bacteria. What's this string-like thing? This looks almost like a cyanobacterium here. This long string-like thingy. Yeah, but where are the protozoans? I saw some paramecia before. Oh, these are all bacteria. Here, these are... Oh, look at this. These are most likely cyanobacteria. Look at this. This guy made a loop. First time for me seeing it. It's kind of... yeah, this is kind of funny looking. Yeah, those filaments here, these are most likely cyanobacteria. And some of them are, as you see, are able to glide and move. But one that's kind of spinning and rotating like this. But a little bit disappointing is the fact, because in the other sample I've seen some paramecia as well. Some, look, a very dense network of it's too dense. Let's move over. Look, some green stuff here. Oh yeah. There is some, yeah. What is this here? Almost looks like some here. Yeah, you see, this might also be a little bit a disadvantage of a high magnification. That yeah, the field of view, of course, is much reduced. But look at those cyanobacteria, or I guess they must be cyanobacteria, because they're in long filaments. Yeah, how they're moving. Occasionally green stuff means it's photosynthetic. Yep, bacteria. Again, yeah. Sometimes that's a nice one over here. You can actually see how they're clustered together, the bacteria. It's almost like a colony. Maybe I suppose that this is actually from the skin, from the surface of the water. Often the bacteria have some kind of a slimy outside layer, glycoprotein and so on. This causes them to stick together and form those biofilm layers. Actually, let's look at this. That's actually quite nice looking here. Look. These are all nicely packed together bacteria. Let's put this one on here like this. Okay, it's a little too... Yeah, doesn't matter. So this is a single layer, seems to be a single layer of bacteria. I think they must be. Yeah. I wonder if it's possible to see them also divide. Yeah. Yeah. I wonder if the biofilm would disturb cross-polarized light. That's a little bit of a problem. It's an interesting question whether those biofilms are kind of optically active. Unfortunately, I cannot do that because I'm using an oil immersion, and I cannot put a filter. I cannot put a filter between the objective and this. Yeah. You see, those things are moving around like worms. They're not worms, obviously. Again, this is again different than from what I saw a couple of hours ago, because obviously I tested this before, because in the live stream I just want to make sure a little bit that I am able to show you some things that where there is something visible. But my specimen from before looked quite different. I wonder what this here at the top is. Look like worms. Some time ago, actually, I made also a time-lapse video of this. This looks pretty cool as well. You just see how then those cells are kind of gliding and moving forward. Look, yeah. There are some of the assuming that they are indeed cyanobacteria, some of them are called oscillatoria because they're oscillating back and forth. Yeah, sometimes you're able to see now the, occasionally you can actually see the individual cells here. Yeah. Sometimes there's some cross sections where you can actually see the individual cells. Yeah, a short question over here. You know what? I'm going to do the following now. I'm going to answer a couple of questions here. Where can I find tardigrades, water bears? I did make quite a few videos on those, so you might want to search for them. You can find them primarily in moss, and they are quite easy to find. If you do not find them, then you have to change a different moss sample. Pick moss that was not that was in the forest, for example. Right, not a moss that is exposed to the heat, for example, on a rock, but one that has been in a shade. I found many water bears there. So if you don't find water bears, then the chances are pretty good that the moss sample was simply bad. Yeah. But otherwise, if you've got a good sample, they're unavoidable almost. Yeah. So I'm occasionally reading again. Yeah. The question is those photosynthetic ones, are they defended by the bacteria? Let's put it this way. I wouldn't say necessarily defended. What you see over here is a complex community, right, of different organisms, bacteria, protozoans, algae, and so on. Yeah. And sometimes there is a codependence. They're not separate. Yeah, of course you can say they are separate organisms, but sometimes they cooperate in certain ways, and they coexist. And sometimes some of them produce nutrients that others need, and so on. Yeah, over here again, a little bit interesting because I could not see any larger protozoans like Paramecia, even though I saw them before. But this could also be due to the fact simply the field of view is too small, and we're just looking at a very tiny area, and for this reason it might be too difficult to see them. But we still have 5 minutes or so. What I'm going to therefore do is I'm going to put a water sample on here without immersion oil, so that you're able to see how this works. Just a second. Notice these slides stick together because I did not dry it properly. So here we go. So I'm just going to show you how I do this. Just like before, what I've done before, I just went into here, directly into the algae over here, and sucked and put a drop on here. And without cover glass, let's have a look at it under low magnification. Let me see the air bubbles. Okay, popping air bubbles lower. Let's see. Some of them are moving so quickly. Oh, a worm. Okay. Here is also one. Yeah. So you see, and this is now in a drop of water. Now when I put a cover glass on top, everything spreads about further, and then it becomes more, it looks a little more diluted. Yeah. Okay, there is another biology question here. How the ecosystem of food chain works inside the jar, why we cannot see one living cell eating the other, maybe just luck. That's one part, and because the food chain in there starts, of course, with the producers, the algae. They do photosynthesis, they produce organic material, and then quite possible that some of them actually decay and break down. And then bacteria and fungi take over. Or in the case of, let's say, those worms, as we see, they are also present. Yeah, of course, they also feed. Yeah, on organisms. And I'm quite sure that, well, not quite sure, I know for certain that many of the things that decompose and break down because of the action of those microorganisms. Yeah, so it is there is a food chain present. But over here, as you can see, I used to see way more. Yeah, but here's one. The problem is if I drop on a cover glass right now, I won't find it anymore, maybe. But we're going to try it as a last little thing. I'm going to try it in any case. I love this switching system where you can switch. It's so much easier. So. Yeah, see, and then of course everything's spread out more. Maybe, yeah, maybe we're lucky. Yeah, see, this one would be nice to look at, but it's so quick that under oil immersion it's probably going to be impossible to chase. The dark circle around an air bubble, that is because of the different refractive index between air and water. So I don't know, maybe we can find an air bubble somewhere. No, let's look at this air bubble over here, right? Yeah, in the center. Yeah, I do that away. You see, actually, there's this dark circle, and that is because the air bubble is not completely flat, but it's round. And there are two different refractive indices of the water and the air, and this causes the light to be diffracted away, and therefore it looks darker right at this at this place. And by actually opening and closing the condenser, you can actually see that by closing the condenser, those everywhere where there's a difference in refractive index, it becomes darker, right? But this can also be a little bit more disturbing because it covers up some structures. Yeah. So that's basically something that the microscope produces, but it can be beneficial because it increases the contrast. So, but let's have a look at some of the. I don't know. You see, this one here would be nice to chase, but if I actually, by the time I've applied the immersion oil, this guy's already gone again. And that's my problem that I have. Over here as well. Yeah, I mean, we can give it a try. Let's give it a try. And what time is it? Oh, okay. And then I'm gonna call it a day. Because 1.5 hours. Yeah. I'll raise it again. You see, and everything is immediately pushed away. Wow, 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 what is this? Is this an amoeba? This looks like an amoeba. This one, this thing right in the center here. Maybe this here as well. Those extensions that you see are the pseudopods. Yeah, two amoeba, definitely. Yeah. Do you see how it changes its shape? Yep. Again, amoeba is a very broad term. If we already talked about taxonomy, it's a so-called amoeboid motion is a polyphyletic trait. That means you find this all over the place. It's not typical to only one. Yeah. Yeah, so quite. I'm quite happy. This was really purely luck. There are two of them next to each other. I've certainly seen larger ones as well, actually much larger ones. But nice over here to see how the bacteria, which you see over here, are significantly smaller, and it's they're actually going to engulf them and then swallow them up. Yeah, yes, there are likely, definitely must be many more around, because if we already see two of them together like this, then the probability must be high that there are a lot of them. Yeah. Up here, yeah, see some bacteria moving around. See? If we already see two of them close together, then probably there are more around. Usually I find them a little bit more difficult to see, because sometimes they don't move quite as quickly, and therefore they're easy to overlook. Yeah. That's a interesting comment. It's also possible, luck, that we found one that just split, hence why they are so close and small. That is possible. Yeah, that's possible. So if you have problems observing, if you don't know if something's moving or not in a sample, take a, what do you call it, a time-lapse image video. Then you will actually see all of the movements much more pronounced. Yep, and here again is part of this, what appears to be surface biofilm. It almost looks pretty. Look at the bacteria here. Okay, ladies and gentlemen, I'm going to do the following. I'm just going to call it a day today, and hope you liked it. And again, a little reminder: do the following. I'll try to make some videos in any case. Yeah. Buffering. Okay, interesting. From my side, the live stream seems to be working at least. Yeah. I wish you, in any case, a nice week, nice 2 weeks. I'll keep you guys then updated a little bit. Maybe I'm going to upload a video or two over the next couple of days, right? And check out also my other channel where I have some more explainer type videos, which quite different in format than this one over here. In any case, I wish you a nice weekend still, and happy microbe hunting as always, and hope to see you again. Okay, bye-bye. Yes, correct. No live stream next week. Okay, because I'm not here. Okay, bye bye.